time. Nice. So for some background of what I pursued during my PhD research experience, I spent the majority of my time with omycetes, Phytophthora and Pythium primarily. But I was also involved in fungicide efficacy testing for some different fungi as well as omycetes, looking at in-field disease management with seed treatments or resistant varieties, investigating the pathogenic host range of Phytophthora sansoniana. And then in corn, I've also gotten some experience with Phylochlorametis in corn, our big pathogen right now, tar spotted corn. While I was at Purdue, though, I worked in a couple different labs. I started in Peter Goldsborough's lab, performing nutrient transfer. I'm just going to switch it all up every 5 minutes. I started in Peter Goldsborough's lab in around 2014 doing nutrient transport in Arabidopsis. Quickly went to Dr. Ames' lab to get some experience in fungal phylogenetics and fungal ecology in Guyana before ultimately making my way to the Purdue Plant and Pest Diagnostics Lab, where I spent a summer and a year getting to know all the pathogens that come in and out of those facilities. So for this seminar, I'm going to start by having a general soybean production and Phytophthora sojae introduction, and then I'll get into Haggis and our package resource that I made for phenotypic data analysis of Phytophthora sojae and other pathogens that interact with plants in a gene-for-gene fashion. I'll then talk about how I used that package to elucidate the Phytophthora sojae pathotype distribution in Michigan, and as well as look at fungicide sensitivity data for that pathogen, trying to look at our management routines and seeing how effective they are currently. Finally, I'll get into this meta-analysis that I've been working on in collaboration with a bunch of different groups here in the United States and in South America, and as well as in Canada. And again, that is looking at resistance gene efficacy over time. So we produce a lot of soybeans nationally. We have around 90 million acres planted, and this brings in around $47 billion to the country each year. In Michigan specifically, we only make up about 2 million of those acres for around an $890 million value. Soybeans in general have many uses. The meal for soybeans can be used in animal feed and human food products. Soybean lecithin is in many different things that we eat. However, the oil can also be used for human food products, energy production such as biodiesel, and then a plethora of other uses such as paint, foam, and shampoo. In terms of global soybean production, the majority of the production occurs in Brazil, followed by the United States and then Argentina, before dropping precipitously for the rest of the world. So our big 3 soybean producing countries are here in North and South America. So some background on Phytophthora sojae, just so we all know what we're talking about. This pathogen is found in all major soybean producing regions. It causes a pre and post emergent stamping off, but its diagnostic feature is really this chocolatey brown lesion that works its way up from the crown of the plant up the stem. And you can see this anytime during the season. So it's not necessarily only a late season. It will usually only happen after the soybean plants themselves have grown quite a bit. For Phytophthora and all omycetes, really an abundance of water is going to increase disease pressure, whether that be from large rainfall events or poorly draining fields or a combination of them both. That free water lets those chemotaxic zoospores to actually swim towards soybean roots and infect. The Phytophthora sojae has a very limited host range, especially when we're considering agronomic hosts, soybean being the only agronomic host. However, you can also find Phytophthora sojae on wild soybean, glycine soja, as well as some native lupine species here in North America. So the Phytophthora disease cycle, I'll start here with dormant oospores surviving in the soil. These dormant oospores will reside in the soil until the environment is conducive for disease. For example, high water or high rainfalls. Those oospores will then germinate either into penetrative hyphae or sporangia, which are full of zoospores. Those zoospores will be released from the sporangia and are chemotaxically attracted to the soybean roots via root exudates. From there, those zoospores will encyst and penetrate the root and begin colonizing the root cortex. During this time, we get reproduction. The oospore and the antheridia attach to each other, and the oogonium is fertilized and turns into our oospore. Around this time, we'll start seeing those classic symptoms: chlorosis, foliar wilting, and that diagnostic chocolatey brown stem canker that we know Phytophthora by. And again, this will continuously occur throughout the season. In terms of Phytophthora management strategies, we have a couple different things at our disposal. We have genetic resistance, both in the form of qualitative or complete resistance. This is our gene-mediated resistance. In soybean production in North America, we primarily see RPS1C and RPS1K, and there are some varieties which may contain the RPS1A, 3A, or 6 loci, but these are not very common and not widely available depending on where you live. We also have a quantitative resistance, which you may have heard called partial resistance or tolerance. This is not a single dominant resistance gene that's conferring this. It is likely many QTLs and genes acting in consortia to build up a tolerance in the plant to the pathogen. In terms of chemical management, we don't really have any sprays that can be used mid-season. So all of our management chemically is typically done at planting, either via seed treatments or in-furrow applications of mefenoxam or metalaxyl, as well as ethoboxam and oxathiopyprolin. Mefenoxam and metalaxyl have been around for a few decades now. It's kind of our gold standard for oomycetes. It works very well on these soil-borne organisms. Ethoboxam is a relatively new compound. We know that it's very efficacious on Phytophthora species, but that some Pythium species are actually inherently resistant to ethoboxam. It's not due to any selection pressure, but actually an evolutionary cause. Similarly, oxathiopyprolin, this is our newest oomycete compound, very efficacious on the Peronospiralian lineages. So Phytophthora, Peronospora are downy mildews, but it is not effective at all on our Pythium species. So seed treatments containing oxathiopyprolin will have an effect only on our Phytophthora species. And Paraclostrobin, this is a general fungicide that's sometimes put into seed treatments that there's been shown to be some efficacy on oomycetes. Cultural practices, we can use tillage or drain tiles in poorly draining areas. Really what we want to do is break up the compaction, especially in the headland areas. And if we have any habitually flooding areas of the field, drain tiles may be useful to mitigate disease. So an example of quantitative resistance, this was a field that we were in Michigan, where the farmer planted 2 different varieties of soybean. Both varieties contained identical RPS genes, and according to the company catalog, had identical company Phytophthora root rot tolerance scores. So they should have equal resistance to the Phytophthora in this field, but obviously you can see that that is not the case. And we've been finding recently that a lot of these field tolerance ratings that are in the seed catalogs that the farmers are actually interacting with and using to identify which varieties they want to plant, they're not necessarily accurate. This is something that we've been trying to work with the seed companies to try and just keep them honest, I guess, try and get them to actually report what the variety looks like when it's in a diseased area. So I've said Phytophthora sojae operates with soybean in a gene-for-gene fashion, and that is via the RPS genes present in the soybean germplasm. So an RPS gene is a soybean resistance gene that interacts with the corresponding effector from Phytophthora sojae. Some terminology going forward, I'm going to talk about pathotypes and pathotype complexity. A Phytophthora sojae pathotype is just a list of genes that isolate or a group of isolates can cause disease on. In terms of complexity, this is going to be the number of RPS genes that an isolate or a group of isolates can overcome. And like I said, these will be important going forward. So looking at this diagram, on the top are our soybean plants, either with an R gene present or an R gene absent. And on the left side, we have Phytophthora, either with a corresponding effector or without a corresponding effector. And what we see is that the R gene present in the soybean germplasm has to be able to recognize and detect the corresponding effector from Phytophthora to be able to mount a defense response and have that hypersensitive response and confer resistance. If the R gene is absent and the corresponding effector is present, then that isolate can cause disease and that plant will be susceptible. Similarly, if we have an R gene present in the soybean germplasm, but the corresponding effector is absent in the pathogen, the plant can't recognize that pathogen is infecting it and will not be able to mount a defense response. So the first project I'll talk about quickly today is Haggis, an R package resource that we made for plant pathologists working in gene-for-gene pathosystems. This is an R package. So if you don't know what R is just a statistical coding language on the computer. And we use it a lot to do data analysis and statistical analysis of our studies. So previously, we were all relying on this Excel sheet that was not very easy to use, and I couldn't even get to work all the way for me. So at our North Central soybean research meetings, there was an identified need that we needed a new way to actually go through and do the data analysis on Phytophthora sojae pathotypes surveys. So I undertook that with the objective of developing an R package that's easy to use and allows rapid reproducible analysis with Phytophthora sojae pathotype data. All right. So we did just that. The Haggis R package uses a very short list of input variables from you that's based on your data collection file. Part of that is a cutoff value, and this is a percentage of plants that were susceptible to the interaction. So if you set a cutoff of 60, as I did here, then that would say that if 60% or more of the plants were killed by the pathogen or were diseased, that would be a susceptible interaction between the isolate and that resistance gene. So using Haggis, we only have 4 functions, and they produce all of the outputs from the Haggis Excel sheet. The summarized gene will give you an R gene efficacy. So what's the number or percent of isolates that can cause disease on each resistance gene? The calculate complexities, this is looking at our pathotype complexity. What's the total number or what's the distribution of pathotype complexity within our sample population? And calculate diversity is looking at alpha diversity indices. So within sample pathotype diversity or within sample diversity. So A, B, and C, these first 3 were all things that were included in the original Excel file. We added also the ability to compare populations. either across time or space, to try and determine significant differences within pathotype compositions and get a better idea of how pathotype diversity operates. So this R package is available for free. It is available to download from CRAN. So far, it has gotten quite a bit of downloads. It has been used in multiple Phytophthora sojae pathotype studies. So we identified the need of having a new data analysis software. We made it and validated it and got it out to the researchers while they were actually doing their data analysis for their pathotype survey, pathotype surveys. Another group saw the use in it and actually used it for Sphaspora sogina, so frog eye leaf spot, looking at resistance gene efficacy in that pathosystem. The one that surprised me the most is it was used in a Parascaris species, a group of nematodes that are parasitic on horses, not anything I know about. But they used some of our code to actually perform the microbiome analysis, comparing different stages of these nematodes microbiomes to each other. And so that was unexpected, but very interesting to see how someone else has come in and used the package for their needs. So Dr. Adam Sparks and Dr. Zachary Noel helped make this package, and this was an open plant pathology initiative. If you are a graduate student or a postdoc that is interested in learning R, but just don't really know where to start, open plant pathology has a lot of great walkthroughs that are relevant to our discipline and based on our discipline, just to help get you started and give you some ideas of how to perform those analyses. I think you should check it out. I think it's pretty interesting and helpful. So using that Haggis package, we then looked at the Phytophthora sojae pathotype distribution and fungicide sensitivity testing in Michigan. So looking at our R gene efficacy and our chemical management options efficacy across the state. Our primary objectives here were to determine effective resistance genes to control Phytophthora sojae in Michigan, what's effective and what's available. And then, like I said, going through and testing our common seed treatment compounds, trying to identify any sort of resistance that could be building in the population to those. If you're interested in getting more than just the spark notes that I'm giving here, this is published in Plant Disease if you wanted to take a look. So this figure is showing the soybean acres planted by county in Michigan. Lighter blue colors would indicate more soybean acres being planted, and then gray is missing data or very little soybean production. The dots that you see indicate sampled counties. If the dots are green, those counties were positive for Phytophthora sojae. Overall, we found between 30 and 35% of those fields that we sampled were actually positive for Phytophthora sojae, and we were able to get isolates out of the soil. So that's from a total of about 70 soil samples across the state. Unsurprisingly, most of our Phytophthora positive soil samples were from the west side of the state, right by Lake Michigan. It stays very wet there. We have a lot of muck soils and heavy clay soils on that side of the state. So we regularly see Phytophthora sojae being a problem there. So that wasn't too surprising. So after we went around Michigan and dug up all this soil, we had to actually bait the pathogen out of the soil to actually be able to use it. So to do this, we used a lengthy baiting process that involves flooding the soil, incubating it in a warm, dark, humid space for 1 to 2 weeks. That lets the levels of Phytophthora sojae grow in the soil. We're providing it a very conducive environment. And after we've provided that conducive environment that allows for bioamplification, we plant those fields or soil samples here with a susceptible soybean variety, something that has no known RPS gene. We use the variety Sloan. I really liked it. Once they're planted, 3 days after planting, remember, we need free water for oomycetes to really infect. So we flood those soil samples, allowing for those sporangia to germinate and release the motile zoospores and allow for infection. And a few days later, what we'll see is emerged plants that are diseased or healthy, and we can preferentially take those diseased plants and isolate them out onto a selective oomycete media. In our case in Michigan, we ended up with about 83 Phytophthora sojae isolates that we used for pathotyping. So what do I mean by pathotyping? When we pathotype an isolate, we take an isolate on the Petri dish and essentially macerate it using a syringe and hypodermic needle. We then use that macerate in another syringe and hypodermic needle to make a small incision on the soybean hypocotyl. These are soybean seedlings, so they're not very big. Make a small incision on the hypocotyl and inject that mycelial agar slurry into the plants, moving forward a infection on those plants. You can see me doing this up here in the top right. I found it very effective to use these long flats where I could plant one line of each soybean variety containing a different resistance gene, so that for one flat, I could have one isolate and it was kept very neat and together. But it does mean that we have to inoculate about 10 plants for 14 different varieties per isolate. So 140 isolates or 140 plants for 83 isolates is what, 10,000? Yeah, 10,000 soybean seedlings. So that's what I did. That's exactly what I did. And here you can see a cartoon representation of the results after we do these inoculations, after we allow those plants and oomycetes to incubate together and for infection to progress. We come back and we have a susceptible variety with no RPS gene. This should always be dead. And then we've seen varying reactions depending on the RPS gene used, if it was efficacious or not. If it was not efficacious, the majority of times the plant would be dead. There were some instances where we did see some leading lesions and not total plant death, and those were still considered susceptible. But what I thought was really interesting was the resistant plants, you can barely even tell they were inoculated by the time you go back. You can see a small scar on the hypocotyl, but otherwise they're still vigorously growing, many times being at V1 by the time we get back. So after doing the inoculations we go through and rate the variety's response, essentially counting the number of plants inoculated and counting the number of plants that were diseased or dead. The last time a Phytophthora sojae pathotype survey was done in Michigan was in 2001. Or it was published in 2001. I think it was performed in the late 90s. And their results were pretty interesting. They found that RPS1A was no longer effective for use in Michigan, but they did find that RPS1B, 1K, 3A, 3B, and 6 were all effective resistance genes up until 2000. When we went through and did our study 20 years later, what we found was pretty interesting. We found that predominantly the entire RPS1 loci was no longer effective for management here in Michigan. Now remember, RPS1C and RPS1K are our two most effective resistance, or our two most widely deployed resistance genes in the US and Canada. So this presents an immediate concern for those Phytophthora positive fields in Michigan. In our case, we found that RPS 3A, 3C, and 4 were still effective in Michigan. However, only one of those is currently introgressed into soybean or commercial soybean varieties, and that's RPS3A. And when I go through and look through the soybean catalog for our maturity groups in Michigan, I can only find one, maybe two varieties in the 2.5 to 3.0 range that actually have the RPS3A gene. The rest of them are either RPS1K or RPS1C, which we believe are really no longer effective in these problematic fields. In terms of pathotype complexity, remember pathotype complexity is the number of resistance genes that an isolate or group of isolates can overcome. We saw a pretty wide distribution in 2001, the previous sampling. When we went back, we saw that those values were now skewed left and much higher than 20 years ago. And indeed, when we go through and look at the means of 20 years ago versus today, we see that on average, these isolates can overcome 2 more resistance genes than they could 20 years ago. So this increases in pathotype complexity present a concern that there are going to be less resistance genes that are actually effective and able to be used within an individual field to successfully manage Phytophthora sojae. So we also did in vitro fungicide testing. We tested mefenoxam, ethoboxam, oxathiopyperlin, and pyraclostrobin. And we did this using a spectrophotometric assay. It's a high throughput assay that relies on optical density to determine relative growth for the isolate. In this visual example, we have 2 isolates, isolate 1 and isolate 2, and 3 different concentrations of mefenoxam from 0, so solvent only in the broth, 1 microgram per mil or 1 ppm, and then 100 ppm. And you can see that isolate 1 is slightly more resistant to or insensitive to mefenoxam than isolate 2. They both have abundant growth in the 0 ppm control, while only isolate 1 is abundantly growing in the 1 ppm of mefenoxam. So this isolate would be less sensitive to mefenoxam than isolate 2. Meanwhile, we see no abundant growth in the 100 ppm, so neither of these isolates are likely truly resistant. This is an example using Pythium, not Phytophthora. So just to get into this high throughput fungicide assay, we grow the omycetes on a low agar percentage Petri dish. In this case, I used dilute lima bean broth with a 0.5% agar concentration. We take plugs from those growing cultures and macerate them by passing them through a syringe and hypodermic needle into a falcon tube. We then vortex that falcon tube to make sure that mycelia and agar is sufficiently homogenized before we pipette them into these little reservoirs that we can then use a multi-channel pipetter with to put them into the 96-well plates. These 96-well plates are where the actual fungicide is amended into the media. So in this case, we use 200 microliters of dilute lima bean broth, as well as a defined concentration of an individual fungicide. So if we're testing 6 different concentrations of a given fungicide, we would need 6 individual plates, 1 for each concentration that we're testing. And then we pipette in the isolates themselves in the same order using just a general 96-well map. Once that master is loaded into the 96-well plate, we incubate these plates for 24 to 48 hours and take readings every 8 hours. So the goal here is to be able to generate a growth curve to identify if the isolate was in the log phase of growth or if it was too late or too early to get an accurate reading of the growth. With the spectrophotometer, we see decreasing OD600s as the fungicide concentration increases. So as fungicide concentration increases, abundance of the pathogen within a well decreases, and the optical density is going to be lower. Once we have this data, we do a lot of data quality control and filtering. So one of those things is called Z prime factor data filtration. So we're essentially taking the growth values we get from the spectrophotometer and ensuring that they're at least 3 standard deviations away from that non-amended control plate. So is there actually a significant difference in growth here that we can rely on to make our relative growth estimates? So as I was saying, we do make a growth curve trying to identify, trying to look at the growth of the pathogen over those 24 to 48 hours. We even take this out later just to make sure we reach that plateau phase. We don't want to sample too early and we don't want to sample too late. We really want to get a good read and sampling on these isolates when they're in that log phase of growth. Once the data has passed those 2 quality control measures, we then go through and estimate EC50 values. So what we found, thankfully, was that there was no in vitro fungicide resistance that we could observe in the sample population. Doesn't mean that it's not out there, but as far as we could sample and identify, we weren't able to find it. So here on the x-axis, we have ethoboxam, mefenoxam, oxadipiperlin, and pyraclostrobin. On the whole, ethoboxam and mefenoxam seemed equally efficacious to Phytophthora sojae. Pyraclostrobin, a broad spectrum fungicide, was also efficacious, but had a more wide range of EC50 values. The highest EC50 value that we determined was still only 0.2 ppm, so very, very small. And we don't take, we think that's a sign that there's no fungicide resistance building from what we could sample. Oxadipiperlin, I know it looks like the data is not here, but it is. We found that oxadipiperlin was actually efficacious up to in concentrations 100 times less than that of ethoboxam or mefenoxam. So oxadipiperlin, very efficacious against Phytophthora species. If I were to do the same assay, and I have, with Pythium species, there would be no reduction in growth. It is ineffective on those Pythium species. So something to keep in mind for the farmers when they're thinking about their disease management routines. So takeaways from this study, we found that RPS 1C and 1K offer little protection here in Michigan, and we were interested to see if that was true in other states or if that was just what we were finding in our sampling. We found that RPS 3A, 3C, and 4 were able to control or were able to effectively manage greater than 60% of the isolates tested. And that pathotype complexity did increase over the last 20 years, at least in Michigan. From this, we think that that means that farmers need to start relying less on those individual RPS 1K and 1C and start looking more into that quantitative resistance or field tolerance to try and get them through until we can get more efficacious resistance genes out. So here's an example of a company seed catalog. In the red box here is the Phytophthora resistance gene. In the green box is the Phytophthora field tolerance. Out of these 18 different varieties here, only a single one has the RPS 3A gene. The rest have RPS 1K, and I think there's one that has RPS 1C and one that has no resistance gene. So we're just kind of troubled by this. Why are we still using these RPS 1K and 1C and making them available to farmers? Advertising. Why are we still advertising these as a resistance management option when we're fairly certain that they don't work anymore? So from our study in Michigan and talks with other states around the US, we started to wonder if this increase in pathotype complexity and loss of the RPS 1C and 1K resistance genes was something specific only to the US, or if this was something that we're observing across countries that actually use these 2 resistance genes for management. And so This study, we were interested in understanding the efficacy of RPS genes over time, trying to identify when their efficacy was waning and when it was actually completely lost for management, at least in the sample population. So to do that, we're looking at national temporal changes in pathotype complexity. So over time in the US, in Canada, in Argentina, in China, how do those RPS genes efficacy change over time? From our study, we saw an increase in the diversity and pathotype complexity of isolates in Michigan over 20 years. We were interested in looking to see if that was actually true for other states or other countries as well. So to do this study, this was my COVID project. To do this study, I transcribed all of the published pathotype manuscripts that I could find into Excel. So taking their summary data tables and putting that as raw data into an Excel file. I then took all of that data for each individual study and validated it using the Haggis-R package just to ensure that the data that I had collected was actually true to what was reported in the manuscript. From there, we went through and identified which nations actually had a temporally available sampling. So did they sample multiple times over the last 20 years, or was there only 1 or 2 samplings over the last 20 years? What data from what countries could we actually use? Once we had that, we further subsetted the data down into the RPS genes that were actually tested across time, making sure that our earliest time point was testing the same resistance genes as the latest time point. Typically, these surveys test between 7 and 14 resistance genes, and they're the same 7 to 14, no matter if it's in the US, Canada, or Argentina. So for this study, we were looking at the RPS1 loci, RPS1A, 1C, and 1K are 3 available resistance genes, as well as RPS3A, and RPS1D, 1B, 6, and 7. These were just resistance genes that were tested across time that we were just interested in having. We took this pathotype data and grouped it by time. So generally, we were looking at the 1990s, 20 to 2010, 2012-ish, and then 2013 up until present day samplings. And then we performed our data analysis looking at efficacy of resistance genes, pathotype complexity over time, and then comparing the composition of those pathotypes over time. So not necessarily part of the study, but something that I'm excited to release. I've taken all this data down from the last 50 years of Phytophthora sojae pathotype studies from 9 different countries. All of this data encompasses 37 different published studies, and all the data that we've taken down has been validated against the published record. And so what this is well, I had a neat little video here. I guess we're not going to see it. This pathotype study, when we get to the point where we're submitting this paper, which we're very close to, we're also going to release the database with the last 50 years of pathotype surveys data taken down. I'm sure it will be helpful in the future for future pathotype studies, but I also think that it could be a very valuable tool in teaching labs, teaching students how to use R, how to look at the data, and just get a good handle on getting used to R. And that's one of the hopes that I really have for this. It was not fun to take down all this data in Excel, but I do think it is a very valuable resource that we can use going forward. Sorry about the video. Oh, there it goes. So we have a bunch of different columns of information that we present, mainly just short survey of what data is actually in there. But then these are all of our different studies that we actually have data for. And when you click on one, it will bring up the validation analyses that we performed on that and show the results so that you can compare it to the published study if you'd like to. And this is just an example of some of the analyses that are performed. So in this study, we used approximately 20 different surveys from the United States, Argentina, Canada, and China. And we used the isolates used for the United States were the most. We typically had a pretty even sampling across time, except for in the China pathotype studies. We were pretty heavy on the back end for samples there. So going back to our objectives, objective 1, pathotype complexity over time. To perform this, we used the Haggis R package to look at this. Objective 2 was looking at pathotype diversity over time. Have pathotypes gotten more diverse, or are they getting less diverse over time? And again, we use the Haggis package as well as principal coordinates analyses to visualize these groups of pathotypes, and using a discriminant analysis of principal components to actually identify what variables in that principal coordinates analysis are driving the differences that we see. Test for significance, permanova, and then beta dispersion to trying to determine if these pathotypes are becoming more similar or more diverse over time. So for objective 1, we did observe or identify that pathotype complexity increases over time. This is something that's been reported in the literature, but there's never been a continental scale study looking at if these things hold true across geography. So again, pathotype complexity, this is the number of resistance genes these isolates can overcome on average. And over time, we did observe that these pathotype complexities increase, sometimes more than others. And in the case of China, potentially just a little bit. So looking at pathotype alpha diversity, so this is within sample diversity of pathotypes, we identified that likewise, those alpha diversity indices were showing that the pathotype diversity was increasing over time. Sometimes there was a small dip or reduction in the middle sampling. I'm not exactly sure why that's the case. I don't have a good explanation for that, but if you have an idea, I would love to hear it. Again, this is just kind of showing us an overview. It doesn't provide specifics. It's just looking at how diverse pathotypes are there. Do we see a lot of one pathotype, or do we see many very different unique pathotypes? So looking at temporal changes over time, we observed that there were very small differences in pathotype diversity over time, gradual increases. And we believe that this is likely due to differences in efficacy of a few genes, but we can't get all of that just by looking at a principal coordinates analyses. So to dive into this deeper, we utilized the discriminant analysis of principal components to identify the resistance genes in each country, which were driving the differences over time. And predominantly what we found was that it was the resistance genes at the RPS1 loci, and particularly RPS1C and 1K in Argentina and the United States were driving differences. In Canada, only RPS1C, not RPS1K, was driving differences. And only RPS1C was driving differences in Canada. RPS1K's efficacy in Canada doesn't appear to have changed a whole lot over time, and I'll show that in not the next slide, but the slide after. So again, going back to pathotype diversity increasing over time, we had some observational evidence that that was the case, but we hadn't performed any statistical analyses to see if they were significant. But both the Permanova as well as the 2-HSD test was able to confirm that these are significantly different from one another. And the mean difference is showing that in general, the beta dispersion seemed to be increasing over time. So using the United States as an example, looking at RPS gene efficacy trajectory over time. So what we're seeing here on the x-axis, I do know that there's no label, but it's an arbitrary time. So starting at 1990 on the far left, working our way up to 2019 on the far right. All of these individual dots are colored, or all the individual dots are pathotype surveys conducted during that time. And we're seeing the average percent of isolates that were pathogenic on that resistance gene. Over the top, we placed a goodness of fit line, trying to identify whether these were increasing over time or decreasing. One of the interesting things that I saw was that RPS gene appears to have been under some sort of selection pressure over time, similar to that of RPS1C and 1K. However, we've never knowingly deployed RPS1B in the United States or Canada. So it's a little confusing to us of how could there be selection pressure if we never actually deployed it. But looking through the literature, we were able to identify some studies providing evidence that RPS1K can detect not only the AVR1K effector from Phytophthora sojae, but also the AVR1B effector, which typically is detected by RPS1B. Likewise, with RPS1C, it is also able to detect that AVR1A loci. So I'm now wondering if this is contributing, this ability to detect multiple effectors from one single resistance gene could be contributing to the longevity of RPS1C and 1K compared to that of 1A. So RPS1C and 1K appear to have remained effective for approximately 30 years, whereas RPS1A seems to have quickly lost efficacy in less than 10. And so looking at the efficacy of RPS genes over time by country, we identified RPS1C and 1K were no longer effective in the United States and Argentina, RPS1C not being effective in Canada, but RPS1K only a slight rise in percents of isolates that could overcome this resistance gene. We identified RPS3A as an effective resistance genes across these locations. And we didn't see a whole lot of selection pressure with the RPS1 genes in China. And that could be because they may not use RPS1C and 1K there. It's difficult for me to try and identify what resistance genes are actually used there just based on the literature. But again, if anyone has any information on that, of which resistance genes are actually used in China, I'd love to know, and please come talk to me. So taking all that into consideration, I made this timeline chart here for the history of Phytophthora sojae in the US, Canada, Argentina, and China. Blue text indicates a first report of the organism in that country. Yellow text is the release of an RPS gene for commercial use. Green would be a loss of efficacy of individual resistance genes, and red would be a waning efficacy. So we start to see the percent of isolates increase on percent of isolates pathogenic increase on that resistance gene, but it hasn't led to a total loss yet. Phytophthora sojae was first reported in the United States in 1948, and then in 1954 in Canada. And approximately 10 years after its identification in Canada, the RPS1A resistance gene was released for use in the United States. This was our first knowingly released RPS gene for Phytophthora sojae management. It wasn't until 1970 that Phytophthora sojae was observed in Argentina. And about 8 years after the first release of our RPS1A in the United States, we see that that resistance is being overcome. This all culminates in about the mid-2010s. We see reports of RPS1C and 1K efficacy beginning to wane in Canada and the United States. And about 5 to 6 years later, our most recent sampling here in the US and Canada, we see that efficacy is completely broken down. And most pathotype studies that were performed recently in various states in the US, we're seeing 80+% of those isolates being able to overcome RPS1C and RPS1K. So takeaways from this project, we did determine that Phytophthora sojae pathotype complexity and diversity increases over time, at least in the countries for which there was data to test that in. We also identified the loss of RPS1C and 1K in 3 of the top 10 soybean producing countries, Argentina, the US, and Canada. And we believe that it's time to look at our other RPS genes for deployment to help manage Phytophthora root rot. So Jingshan Ma here at Purdue his group recently identified RPS11, which looks like a very, it looks like a very good resistance gene to deploy, I guess. They tested a great diversity of pathotypes against the resistance gene, and it was able to protect against 80+% of those isolates. And that's pretty good. In the short term, RPS3A may be a good candidate for redeployment. It's already introgressed into soybean commercial varieties. We just need to make it more widely available because this gene, not only in Michigan, but also in surrounding states in the US and Canada, we're seeing that it is at least reported to be effective against those populations from this study. And similar to what we found in Michigan, we do believe that quantitative resistance or this partial resistance will be even more important going forward to help manage our Phytophthora stem and root rot problems. So that was my stuff that I was going to talk on. I also just wanted to say, just to kind of show everybody what I've been up to since I've been gone. My dissertation projects were primarily around resistance gene efficacy, fungicide sensitivity, and molecular diagnostics for omycetes. I've also done some work on Phytophthora sansimiana, looking at host range characterizations. This is a Phytophthora species that infects corn, soy, dry bean, fur. I mean, it has an extremely wide host range. We were just trying to better characterize that using a group of Michigan specific crops. In that same turn, I've worked on a nanocorn Illumina genome for Phytophthora sansimiana in the hopes that we can try and identify some very conserved effectors that are used again across host species, and then also potentially some species specific effectors. Pythium, so in terms of Pythium species, I'm also working on a Pythium unsimulatum. This is a lettuce wilt pathogen that is a big problem out in the Salinas Valley right now. Pythium unsimulatum and Patient's necrotic spot virus are the reason that your lettuce is a lot more expensive more recently. So we're hoping that that rapid diagnostic tool will actually be able to help predict risk in a field and help farmers better determine what their management routines should be. I've also worked on tar spot of corn, doing first reports and doing an early microbiome study, trying to identify just which fungal entities are actually involved in tar spot of corn in Michigan. And that was pretty interesting. One of the favorite things that I do is working with the soybean breeders at Michigan State. So they love it when I characterize Pythium and Phytophthora isolates for virulence and aggressiveness across species. And they like to take those isolates and then use them to identify QTLs for resistance, or in the case of Phytophthora sansimiana, we believe we found the first R gene for Phytophthora sansimiana in some of those soybean lines. So I'm really excited about that, and I hope we get that out soon because I think it's really cool. And last but certainly not least, some experience in tropical mycology with Dr. Kathy Aim here. I was lucky enough to go with her to Guyana and collect aerial litter trapping fungi. So these are fungi that live in the canopy of the rainforest and trap litter as it falls to the understory, and they colonize it and degrade it. And it's essentially like a second layer of degradation going on separate from the understory. Anything that's in bold and underlined is published and you can find online. So acknowledgements, the Children's Lab obviously is where I did my PhD and I'm currently working on my postdoc. And then all of my long list of collaborators who have made this work possible. Thank you very much. And my funding agencies as well who keep us going. And with that, I'll take any questions. These are some of the wonderful fungi that I found around Michigan, as well as my Nokia computer, as well as the mycoheterotrophic ghost pipes. And these are a plant that produces no chlorophyll, but feeds off of fungi in the soil. And it's always a treat to find them. Thank you.
Well, Austin, we have a few minutes for questions. I don't know if you have any online that we would not dare you to ask it.
Yes, sir.
This whole story, of course, closely parallels the history of rust and powdery mildew resistance breeding and cereals, but this is a slower process because you're dealing with soil-borne fungus. But my question is, I haven't followed the literature since I retired, but there was an idea that we used to call them virulence genes that are now these effector genes because they appear to be null alleles. If an organism, a fungus has too many of them, there's a loss of fitness.
Yeah.
And so the idea was if you could stack enough R genes into a cultivar, the race or a pathotype might be virulent, but it's such a weenie, it couldn't do anything.
Yeah, it wouldn't be able to cause much disease.
Has there been any headway on that idea? Do you know?
Not in soybean that I know
Because
of.
you've got a lot of pathotype complexity. That,
Yeah.
8 or 9 genes that are overcome.
Yeah, not that I know of in soybean. I know that there was just a paper released recently. I forget which crop it was. It was some grass casing, where they did stack resistance genes for a long time, and they found that the pathogen just ended up overcoming both of them, and it wasn't as useful as they thought it would be. Whether that'll be the same for Phytophthora sojae as a soil-borne organism that's not as prolific and easily spread, I don't know. But there's a lot going on with Phytophthora sojae. It can have these genes in its genome and just have them silenced so that it's not being expressed, and that's as good as not having it at all in terms of pathogenicity. But there's a lot of work going on trying to kind of untangle what exactly is going on and how these pathotypes, what is selecting for the change over time. I think based on my data, part of it is due to the selection pressure we use with resistance genes. That's been contested in the past, and there has been some data from Iowa showing that pathotypes change even when soybeans aren't planted. So if they inoculated with a specific pathotype in the first rotation of soybean, they go to corn and then back to soybean, those pathotypes have changed during the corn rotation. So it's not very straightforward, unfortunately.
Still no questions online. Any other questions?
Just give one. Oh, go ahead.
I was just going to add a comment more than anything else. So thanks, Austin, for your presentation. I actually work for Jinshin Law downstairs, and just wanted to add as a comment that along with RPS 11, which was recently published, we are working on 3 new novel R genes for soybean Phytophthora sojae resistance. And one is RPS 14, and it's been published. Or the rough mapping's been published, but we're going to continue working on it. But they have a resistance to a wide variety of isolates. So,
Okay, all right, that was my question to you.
they should be commercially deployed to track
Do you know where they are in terms of deployment, or are they still getting introgressed? Are the
We're
markers
still in the
there?
process of characterizing them, but in the next couple of years, they could be visible.
That would be awesome.
I just want to briefly mention, a lot of the genetics of this soybean resistance was done here at Purdue.
Mhm.
Kirk Aptow and Francis Laviolette, once you first found it. But a guy named Herb Mueller did his PhD here in the early 70s. And did a beautiful genetic study of resistance in soybeans. Published in Phytopathology, I think, and it's worth looking at that paper, even though it's old.
That doesn't mean anything. Who says his name was Herb Miller?
Herb Mueller.
Mueller.
M-U-E-L-L-E-R.
I'm gonna write it down. so I
Unfortunately, I
don't-
think he died several years ago. It's a beautiful study he did.
I'm not super familiar with that, but I would love to read it.
Austin, do you find that is Sansomiana being a root nibbler or anything as far as what the interaction is? Does it make the plants more susceptible to the?
Yeah,
sojae?
I would
What's the
love to do
interaction?
some co-inoculation studies. From my experience using it in the growth chamber and in the field, I've done some inoculated field studies. I'm seeing it do more of a pre-emergence damping off or a very early post-emergence damping off. But when I see mature plants in the field, I'm typically finding it on the taproot.
Right.
So Phytophthora sojae, we see a lot on like those feeder roots, but Sansomiana, I'm seeing more on the tap, but I haven't really been keeping track either, I guess. It looks incredibly virulent when we do these growth chamber or seed rot assays, much more virulent than Phytophthora sojae. And so that's part of the reason we're so interested in it. And likewise, we're seeing it pop up more in these new Phytophthora surveys now that it's been differentiated from the Megasperma species
Well,
complex.
and that was my next question is, so we come up with these other genes that are going to be resistant to sojae, but is anybody looking to see if they're also
Oh, I did do
resistant
that.
to Sansomiana?
So I did test 15 Phytophthora sansomiana isolates to our sojae resistance genes, and none were replicably useful. It doesn't appear that any of those Phytophthora sojae RPS genes will be efficacious for Phytophthora sansomiana, at least from what I've tested in Ohio. It's part of the reason I'm so excited about this potentially novel R gene for Phytophthora sansomiana. I think it'd be interesting.
All right, well, I think we're out of time, so I thank you again.
It's done now. It's done.
Okay, so he's here for a
Dinner.
little bit. Very similar.
Yeah,